today they're going to tape this, and so I'll try to get the lecture done sort of on time. And today's lecture is not as hard as the last one. But then I'm very happy to schedule at least maybe 2 review sessions. So, anytime you have time to get together, we can do that. So I think we have to wait probably a few more minutes before we get a quorum here. We need a few more people to show up. So everyone found the last homework set a little difficult, maybe? Is that, no? Not so bad? It was a little difficult? Okay, yeah. There are a lot of, last lecture there were a lot of sort of abstract concepts that I had to introduce. And again, if we do this in a sort of informal setting, and just you can ask questions and I can answer them, I think it'll be much easier for people to understand that material. I tried to leave out some of the more difficult material from the homework assignments. I hope you noticed that. A couple things that were on slides I didn't actually cover in the lecture, and those I didn't include in the homework either. So, and it's only the homework that you need to really master for the final exam. So how about other classes that you've been taking? Do you get homework assignments in most of your classes, or not? But how about just regular classes? Once in a while? So it must be very hard to know what to study exactly, right? Are you told that you have to only know the material in the slides, or are you referred to a textbook to learn? Both? Pardon? Uh-huh. Yeah. Yeah, because there's so much to learn. It really is hard to know. For example, reading the genetics textbook, there's so much information. There's too much to expect to learn on first reading, that's for sure. Yeah, don't close the door completely, because there'll be people in probably. So there's no consensus yet, or no? Voted? Do you want to vote? I guess. Or some idea, some.
I think the dates you suggested were great.
So like next Friday, a week from today, would be a good time. Your Friday mornings are free, right? Yeah. Instead of lab? Oh, that would be fine, actually. Yeah, that's fine. Yeah, okay. Sure. What time on Wednesday? 1:30? Okay. So how about next Wednesday at 1:30? And we'll plan on maybe 2 hours or so. And it'll just be informed. You can just ask me questions, and I'll try to answer them. Yeah. I think there are 2 more, if I remember the schedule. Yeah. There's going to be, next week will be, there are at least 2 more. There'll be genetics of cancer next week. And it should be a very good lecture. This guy's really a good lecture. And then there's going to be a lecture, I think, on developmental defects. Yeah. Yeah, so 2 more, I'm pretty sure. Well, it's up to you. I mean, we can hold one, and you can decide if you want another one. How's that? Pardon? Okay, so I guess pretty soon we better get started. There are a significant number of people who are not here yet, but okay. Yeah, okay. That's it, okay. Okay, so maybe we'll get started today then. So this is the fourth of 4 lectures that I've been asked to give you, covering the genetic basis of various human diseases. The first lecture we covered diseases that had to do with abnormalities of the human karyotype or structural changes in individual chromosomes. The second lecture we talked about Mendelian disorders, which are primarily caused by single mutations that have high penetrance and expressivity. And then last week we had, I think, a rather challenging lecture on complex disorders, where these are the disorders that most of us are going to be faced with in our personal lives and as doctors that you'll be having to diagnose in the future. And the genetic basis of this is very complicated, of these disorders. There are usually many genes that are involved, and also environmental factors as well. Today we're going to talk about diseases that have to do with the mitochondria, and in particular we're going to concentrate on diseases that result from mutations in the mitochondrial genome. So the topics will then begin with sort of the biology of mitochondria and the mitochondrial chromosome. The biology of mitochondria, I think you covered it probably in the last class. So we'll go over that just very briefly. But we'll spend some time analyzing the mitochondrial chromosome and talk about the genetics of mitochondria and mitochondrial disease. And then some, I hope, interesting sort of topic about what we can learn about the history of the human population, the current human population, from studying the genetics of mitochondria. And then finally, we'll talk about some mitochondrial diseases that have been well studied. And as I'll mention later, these diseases are all very rare. And so it's unlikely that as physicians you're going to encounter these. You may encounter them once or twice in your career. But they're important to know about because they are among the first diseases where the molecular cause of the disease was very clearly defined. And furthermore, collectively, mitochondrial disorders probably account for a significant fraction of pathology that you'll run across as doctors. A lot of that has not been discovered yet, has not been elucidated, but over the next few years, especially as the genome is analyzed and the genome of many, many individuals is analyzed in more detail, more and more diseases, I think, will be shown to have a link with mitochondrial abnormalities. So this class of disease, I think, is going to be very important for you as clinicians in the future. Okay, so as an introduction, all cells in the human body, except mature erythrocytes, contain mitochondria. Somatic cells differ in the number of mitochondria that they have, but they're usually a couple hundred to several thousand, depending upon the need for the cell for ATP. The energy requirements for cells like skeletal muscle, cardiac muscle, hepatocytes, and neurons is very high. And so these cells tend to have larger numbers of mitochondria. And these tissues are also, therefore, more susceptible to mutations that affect mitochondrial genes. Sperm, on the other hand, contain only a few mitochondria. And as I'll explain later on, these are usually lost upon fertilization, so that the mitochondria that we all possess in all the cells of our body derive almost exclusively from our mother. Okay, as you discussed in the last course, the cellular functions of mitochondria include the synthesis of acetyl-CoA, and then the oxidation of acetyl-CoA via the Krebs cycle, or tricarboxylic acid cycle, to yield the reduced compounds NADH and FADH2. And then using that reducing potential, ATP is synthesized by the mitochondria. And so the mitochondria are sort of the batteries, the powerhouse of cells, and are the chief source for ATP for cellular use. Mitochondria also carry out the oxidation of fatty acids and amino acids. And I think you talked about apoptosis last semester as well, or last class as well, I hope. This is programmed cell death, and mitochondria play a very important role in the regulation of this programmed cell death. You'll probably hear more about this next week when we talk about genetics of cancer. And then they're also important for the sequestration of cytoplasmic calcium. As you probably know, the concentration of calcium inside cells is very low. It's on the order of 100 nanomolar to, at most, 1 micromolar. And whereas the concentration of calcium outside the cell is in the millimolar range. So there's a huge difference in concentration of calcium. And it turns out that high intracellular calcium, or intracytoplasmic calcium, is very bad for cells. It usually leads to cell death. And so cells have a variety of mechanisms for sequestering that calcium, for storing it in a safe compartment. And the mitochondria is one of those compartments. On the other hand, in addition to these sort of constructive functions that the mitochondria play in the cells, they're also the source of free radicals. And the free radicals, as you know, are very highly reactive chemicals that can react with proteins and DNA, for example, and cause mutations or cause other malfunctioning in the cell. And so they're also a source of problem, especially if there are mutations that affect the electron transport. You're probably familiar with the structure. The mitochondria, as I'll explain in more detail in a minute, are probably derived from free-living protozoa, but have over the over 1.5 billion years of evolution, become changed from a symbiotic protozoan-like organism inside cells into a cellular organelle. And this organelle has two membranes, an inner and outer membrane, and then there's a space in between. And each of these compartments has important functions. In terms of this is a slide that shows the topology of the mitochondria. And so the space in between the two membranes is an important compartment for storing calcium, for example, for protecting the cell from too high concentration of calcium in the cytoplasm. Whereas the inner compartment, the matrix, is important for most of the biochemical processes that take place. And it's also the site of where the mitochondrial DNA is located. Now, the mitochondrial genome is very small compared to the genome of the entire chromosomal-based genome. It contains only about 16,000, a little over 16,000 base pairs, and encodes only 37 genes. And all of these genes are essential for mitochondrial function. Together, the genes comprise about 93% of the mitochondrial genome, and the remaining 7% is non-coding DNA. Unlike most genes in the chromosomal genes, mitochondrial genes contain only exons. There are no introns that are present. There are also no histones that to form chromatin. 13 of the genes encode proteins, enzymes that function in oxidative phosphorylation, and the remaining 24 genes encode 2 ribosomal RNAs and 22 transfer RNAs. All the other additional proteins, and there are about 3,000 or so that are found in mitochondria, are derived from the nuclear genome and have to be imported into the various compartments of the mitochondria. This is a picture of the structure of the mitochondrial genome. And it's unusual in that it has some unusual features. First of all, it's a closed loop, like you often find in bacteria. But it has one part of it there's a triple helix that is formed, where this portion of the chromosome is replicated twice, and one short segment of that hydrogen bonds to this inner strand, and the other one is sort of free. And this is called the D loop. The two strands are different in their composition. The inner strand here is the light strand, and this is rich in cytosine residues, where the heavy strand is rich in guanosine. So these can be separated on density gradients, for example. Let's see. So the replication of the chromosome takes place inside the mitochondrial matrix, and this is important. It's catalyzed by a DNA polymerase gamma. So it's a different DNA polymerase that is used for the replication of the chromosomal DNA. But this is encoded by a chromosomal gene. So there are at least several copies, maybe at least 5 or 10, are present within each mitochondrion. And as I just mentioned, there are no histones that bind the DNA. Okay, so here's a more detailed map of the arrangement of genes. And most of the genes are encoded by the heavy strand, and their positions are shown along this circle here. 6 of the 7 protein encoding genes are encoded on that strand, and 1 is encoded on the internal strand. And the ribosomal RNAs are also encoded on the outer heavy strand. There are 2 promoters that promote this transcription. 2 are promote here for the heavy strand, and then there's an additional one for the light strand. And unlike transcription of genomic genes, but very similar to what you see in bacteria, these transcripts, there are 1 or 2 very long transcripts that are made, and then the mRNAs are derived by cleavage of that transcript. So as you know, there are very, very few examples of chromosomal genes which are transcribed as a long transcript and then cut up. This is very common in bacteria, however. What you can see then, this is color-coded, so the protein-coding genes are in blue, right here. And the RNA genes, the 2 of them are right here, are pink. And then the genes encoding various tRNAs are shown in this purple. Okay. Now, this is another picture. I'm going to show you, later on, I'm going to show you this again. This shows you sort of more clearly where the different genes are located. And later on in the lecture, I'm going to show you this again and show you the positions of various mutations that cause human disorders. Unlike the human genome, where genes that encode RNAs or proteins comprise only a very small fraction, probably less than 3% of the entire genome, most of the genome of the mitochondria is used for coding something important, either a protein or a type of RNA. And it's so compact that there are even examples where there's an overlap in the reading frame of the encoded proteins. So, for example, this is the mitochondrial ATPase subunit number 8 gene, and it starts over here and it has a continuous reading frame like this. And it overlaps with the mitochondrial subunit 6 ATPase gene. So there's an overlap in the gene. And furthermore, the stop codon for this gene is missing. It's not encoded in the genomic DNA, but rather depends upon a polyadenylation of this site to complete the stop signal. So this is the result of very stringent selection, probably over 1.5 billion years, that have reduced the chromosome of what was probably originally a free-living organism, free-living bacteria-like organism, to the most sort of compact form that's possible. Okay, so this is just a summary. Yeah.
What's the polyadenylation?
Oh, you didn't study that in your last lecture? This is very important. Okay, we'll take a brief digression here. Yeah, this is very important. You should have covered this last semester. When genes are transcribed, the RNA is synthesized from the 5' end of the gene to the 3' end, and a transcript is made. And it's a very long transcript. And as you know, it contains both introns and exons. And these are eventually, the introns are removed by splicing, and so the coding sequences are all brought close together. And then at the very 3' end of the transcript, there's a signal for a nuclease to cut the mRNA transcript. And so it cuts 5' to the end of the transcript. And there's an AT-rich sequence that acts as a binding site for that nuclease. And once it's cut, there's a 3' hydroxyl group that's exposed. And then there's an enzyme called terminal transferase that adds many A residues to the end of that. And these extend, they're usually several hundred, 200 or 300 often. And without the addition of this poly A tail to the mRNA, the mRNA is not stable. It's first of all, it's not transported to the cytoplasm very effectively without this. And if it gets into the cytoplasm and it doesn't have this poly A tail, it tends to be very unstable. So that tail, the addition of that is called polyadenylation. Okay, and that occurs both in the for RNAs that are transcribed from the genome, chromosomal RNAs, as well as for mitochondrial RNAs. Okay, does that sound familiar?
I feel like I learned it a while ago.
Oh, okay. Yeah, okay. Yeah. Okay, so then this is sort of a summary of what I've just said. These are the various subunits of the oxidation phosphorylation system that are encoded by the mitochondrial genome. And as you can see, this is only a small fraction of the proteins that are needed to make a functional oxidative phosphorylation system. The remaining ones are encoded by the nuclear genome. By contrast, however, the RNAs and that are required for translation of proteins are all encoded by the mitochondrial genome, whereas ribosomal proteins come from the nucleus. Now, I told you just a minute ago that there are 37 genes encoded by the mitochondrial genome, but there's one possible addition. This is still a little bit controversial, but recent evidence has suggested that there may be a 38th gene that's encoded in the it encodes a short amino acid containing 21 to 24 amino acid, a short peptide containing 21 to 24 amino acids. And this apparently is found within the 16S ribosomal RNA. And this again is, I think it certainly has not reached your textbook yet, and it's a little bit controversial, but this seems to have anti-apoptotic properties, especially with respect to the protein that is one of the causes of Alzheimer's disease, the APP protein. So this is something that you may read about more in the future. Okay, so the mitochondrial genetic code is very similar to the genetic code that's used by chromosomal genes, but there are some subtle differences. First of all, 60 codons are used to specify amino acids compared to 61 in the nuclear genetic code. There are 4 stop codons instead of 3. And although in the human genome, apparently these 2 are the ones that are used. But in other vertebrates, these are also used. So the nuclear stop codon UGA encodes tryptophan in mitochondria, so that there are some differences here. And AUA encodes methionine rather than isoleucine. So there are actually, in addition to AUG, there's AUA can initiate protein synthesis. Other organisms, by the way, have other differences in the genetic code. And the idea here is that the genetic code is pretty much universal for chromosomal genes, for the nuclear genome. But here, there are fewer genes, and the idea is there may be a little more flexibility in terms of evolution. There may not be as stringent a selection for the maintenance of the original genetic code in mitochondria, because in various organisms, if you look at the genes that are encoded on chromosomes, the standard genetic code is universal. But if you look in the mitochondria, there are subtle differences between organisms. Okay, so the RNAs are an interesting story as well. There are fewer tRNAs that are available for mitochondria. 18 of the 20 amino acids that are put into proteins couple to a single mitochondrial TNA subtype. 2 amino acids, leucine and serine, each couple to one of 2 different subtypes. So in the nuclear genome, there's a larger number of these subtypes of tRNAs. This gives a total of this accounts for the 22 mitochondrial subtypes. So together, the 22 mitochondrial tRNAs are able to recognize 60 mitochondrial codons that specify amino acids by exploiting the third wobble base in the codons. Now, I don't know if you should have covered this last semester as well, but I'll go over it again if you don't remember it or it wasn't introduced. And the wobble base is found in the third position of the codon. So, for example, these all in mitochondria, each of these 4 sequences, CCA, CCT, CCC, CCG, all encode proline. But there's only one mitochondrial tRNA that recognizes all of these. Now, in order to, I'll explain the molecular basis for that in just a minute. But this is, again, an example of sort of the economy that mitochondria demonstrates. Instead of having 4 different species of tRNA, that one to recognize this codon, another one here, another one here, another one here, evolution has produced one species of tRNA that recognizes all 4. So among the tRNAs, 8 mitochondrial tRNAs recognize 4 amino acids like this that differ in the third position, and 14 recognize 2 amino acids, so 2 different codons. And so if you put that together, that accounts for all 60 codons that are recognized. This is just a chart of the vertebrate mitochondrial DNA codon. You can look at this in more detail at home. So so this is the structure of the tRNA, and as you probably learned last semester, the tRNAs contain a variety of chemically modified bases. For example, there's a so-called pseudouridine base that's found in tRNA that's not found in messenger RNA. And so this has the mitochondrial tRNAs have all those normal modifications of tRNA, but in addition, they have a unique modification involving this position right here. So this is the position that will pair with the so-called wobble nucleotide of the mRNA. So this A, T, C, or G would be the residue that would hybridize to this residue right here. So this is the first residue going from the 5' to 3' end here. And it turns out that this residue is chemically modified. It contains a taurine molecule or a sulfonated form that's shown right here. So it contains one of these two. And this chemical modification changes the binding property of that nucleotide. So it now will recognize any one of the four different opposing nucleotides that are found in the codon. And it turns out that this modification is absolutely essential to the normal functioning of the mitochondria. I'll go into this again later in the lecture, but there are mutations, for example, up here or there, that change the conformation of the tRNA. And by doing so, this chemical modification from taking place. And so people who have these mutations cannot make tRNA molecules that cannot recognize the different choices in that wobble position. And because they can't do that efficiently, they can't synthesize the proteins that are mitochondrial proteins that are needed to make ATP. And so it's a source of disease. So this was a very surprising discovery that was made fairly recently. But it's a demonstration, this is a clear demonstration of a molecular cause for a human disorder. So this model was surprising to find, and it's also surprising, it's also essential for the functioning of mitochondria. Okay, this is just another picture of this. The tRNA that carries leucine, for example, and recognizes this UU, and then this is the wobble position here. It has this modification of this first U here. And if this is mutated, if there's a mutation that prevents this from being formed, then this disorder, which we'll talk about later in the lecture, the MELAS disorder is the result. And likewise, if this chemical modification of this U does not take place, then this disorder is caused. And if there's a depletion of taurine in the diet, then there's a intermediate structure that is formed here, but the taurine is missing, and that also leads to human disorder. So this is a very important chemical modification. Yeah. Well, remember they bind in an anti-parallel way. Yeah, so that's why. So the last position in the mRNA is recognized by the first position of the tRNA. Okay, so then just to give you another example of disorders that can arise from mutations in mitochondrial proteins. These are different pathways that for the metabolism or synthesis of DNA. And the ones that all of these are encoded by genomic genes. And I just want to, and each of these, when they're mutated, is the source of some sort of mitochondrial disorder. I just want to call your attention to the DNA polymerase gamma, which is shown here, because this has been particularly well studied. And so this complex contains the gamma polymerase itself, which is the catalytic subunit of the polymerase. Then there's an accessory subunit that's encoded by another gene, and then a helicase a helicase is an enzyme that helps to release the tension in twisted RNA or supercoiled RNA. And mutations in each of these subunits has been linked to human disorders, and in particular, mutations in the catalytic subunit has been linked to many different types of mitochondrial disorders. So again, today we're going to concentrate primarily on disorders that result from mutations in mitochondrial encoded genes. But keep in mind that mitochondrial disorders can also arise from mutations in genes that are encoded by in the chromosomes. Okay, this is just a picture. This is a catalog of many, many mutations that have been identified in that DNA polymerase gene. And you can see there are many. All of these are very rare, but looking at many, many thousands of patients, these have been identified. And depending upon the location of the mutation, the severity of the disorder and the phenotype of the disorder is different as well. Okay, so this diagram shows the proposed endosymbiotic origin of mitochondria. And I must say that when I was in college and graduate school, this was still considered a very controversial topic. This was heatedly debated. But it turns out that the preponderance of the evidence now favors the idea that mitochondria were originally free-living type of protozoan and had then colonized, probably through some sort of symbiotic relationship, colonized another type of cell, and then over a very long period of evolution, lost the ability to live free of the cell, and the cell itself became sort of dependent upon this symbiotic organelle. Chloroplasts have been proposed to also derive. Chloroplasts in plants are now thought to have been formed by the same process. One of the interesting aspects of the fact that I mentioned this at the beginning of the lecture, but all of the mitochondria that we have in all of our cells were inherited from our mother. Sperm contain a small number of mitochondria, but these are either lost during the process of forming the sperm, or even if that somehow comes into the oocyte that's fertilized, those mitochondria tend to be degraded. So almost all of the almost with 100% certainty, mitochondrial genes are inherited from one's mother. And because of that, you can trace back in time, in theory, the origin of every single mitochondrial chromosome to some ancestor in the remote past. And by looking at different mutations that different individuals in the current population, different mitochondrial variants that exist in the current population, geneticists can actually construct a tree showing roughly when in time, different variants appeared in the human population. And from that, construct a tree like this, tracing back to, in theory, a single individual. So in theory, every one of us contains mRNA that can be traced back to one woman in the remote past. And the estimate now is that this woman lived in Africa. There are some maps that even show a place where they think this woman lived. And this woman lived roughly, in this time period, up to probably no longer ago than 200,000 years. Similar maps can be made for the Y chromosome. As I explained last time, the Y chromosome is inherited strictly through the male line. And by looking at different genetic variants in Y chromosomes that are present in modern populations, it's possible to construct a tree that's very similar to this. And for the Y chromosome as well, every male contains a Y chromosome that is ultimately derived from one individual who lived in Africa roughly at the same time as this, roughly within a few thousand years. So when this was discovered, I think in the 1980s, if I remember right, and this was dubbed sort of the mitochondrial Eve. And so the man who produced all of the Y chromosomes would be the mitochondria, or the Y chromosome atom, I guess. But they certainly didn't know each other, and they probably, they lived roughly in the same epoch, but not at exactly the same time. So because of this, then, this looking at variants in the mitochondrial DNA yields an enormous amount of very useful information about when different populations, say, moved out of Africa. And it can also be used to show that certain populations moved out of Africa and then later moved back into Africa. And so if you look at a world map of these variations, different people in different parts of the world contain characteristic spectra of these variations. Okay? So this is a rather recent development, and this is still a very intensely researched area of research, and more detail is being discovered about the origin of the modern human populations based on this analysis. Now, you may have read about this in either the tabloids or on Google, but this is also, this past year, this was also in the news again. How many people read about this? The discovery of the skeleton of Richard III? You remember that? Has anyone read Shakespeare's play Richard III, or heard of it? Shakespeare wrote a historical play called Richard III, and in which Richard is an incredible villain who murders his two nephews, which is probably true. He's a hunchback and just a ruthless individual who finally met his demise on the field of Bosworth in 1485 at the hand of Henry Tudor, who became Henry VII. Henry Tudor was a descendant of the Lancaster branch of the royal family, where Richard III was the descendant of the York branch. And the York branch and the two branches of the family had been fighting for generations. And the battle that was held in 1485 was the last battle of the so-called, this war between the two branches of the royal family, called the War of the Roses. It's the last war, last battle. And in this battle, Richard III, apparently his horse became bogged down in thick mud. He fell off his horse, and then he was killed by the soldiers of Henry Tudor. And this in Shakespeare's play, he has Richard III falling off his horse and saying, a horse, my kingdom for a horse." These are famous lines from Shakespeare's play, and apparently this is actually based on some fact. The king was then stripped naked, thrown on a horse, and according to legend, either dumped in a nearby river or dumped into a well. At any rate, no one knew what happened to him until a year or so ago, when some archaeologists working on a historical hunch decided to look at in the courtyard of what was once an abbey, of Catholic abbey that was located near the battlefield. And they had guessed that maybe his body had been taken there, and maybe the monks in the abbey had buried him there. Currently, this is all under asphalt. It's part of a parking lot. So they dug up the parking lot and started excavating. And lo and behold, they found a body that was right from the right historical period. It had shown, had been hastily buried. It showed scars from battle that were probably lethal. And to clinch it, the spine of this individual had scoliosis. So he wasn't the hunchback that Shakespeare had imagined, but he definitely had a curved spine. So putting this all together, people were pretty sure, historians, that this was actually the missing body of Richard III, discovered 500 years after the fact. Well, this was a couple years ago when all this was done. And then this year, the geneticists succeeded in extracting DNA from the bones. And what they did was they tried to match, they were able to sequence the mitochondrial DNA and also the type do genotyping on the Y chromosome. And what they tried to do then is to match see if there was a match between this mitochondrial DNA and two living descendants, the descendants according to the historical record, to see if their mitochondrial DNA matched. Now, as I mentioned last lecture, there's something like 19 or 20 steps, generations between people living today and Richard III. And so if you try to look at the autosomal DNA to look for a match, you wouldn't find anything. Because with every generation, you lose half of the DNA. So this would be, the shared DNA would be less than 1 over 2 to the 20th or 2 to the 21st power. It would be a very, very small fraction. The mitochondrial DNA, however, if you trace it through the maternal line, could be expected to be inherited with almost no changes, even after 500 years. And so they found two descendants, according to the historical record descendants. And they found there was one individual had an exact match with this mitochondrial DNA. And another individual had 1 point mutation. So this is almost a perfect match. However, when they looked at the descendants through the paternal line, again, the historical descendants, they didn't find any matches. None of these guys. The Y chromosome did not match this at all. They did find a very good match with another presumptive historical descendant, and that is Henry Somerset, who lived during this period. And here, 4 of the 5 individuals matched his Y chromosome exactly. So the conclusion is that these two people were not related to each other. Actually, historically, they're supposed to be related to each other through the male lineage, but they're actually not. And what you can conclude almost conclusively is that there was some usurper here. There was some infidelity in the royal line, some midnight tryst or something, that resulted in a Y chromosome, a foreign Y chromosome being introduced into the royal lineage. Okay? And so this is what this guy had inherited, and these guys have it, but it doesn't match with him. So this was a big surprise and sort of scandalous, even though it probably occurred sometime during 500 years of history. But apparently, the geneticists were actually not very surprised by this at all, because apparently, on the average, there's about a 1% to 2% occurrence of this in every generation. If you look at the entire population, it's fairly frequent. And this is actually, as we enter the age of genetic testing, this is something that's going to be found more and more frequently. Already, for example, when geneticists do in the population, yes, and it's already been found. Actually, the geneticists, in addition to doing case control studies, often construct trio studies where they genotype the person with the disease and then both parents, and not infrequently, fairly frequently, they find that the father is really not the father of the proband. And the geneticists, of course, sort of discreetly remove these from the data set. But as genetic testing becomes more and more wide, this is going to be more and more of these incidences will be discovered. This is another very interesting topic. It turns out that infidelity in the animal kingdom, especially among birds, is extremely common. And so maybe we shouldn't be surprised that it's common among humans as well. But this is a sort of a famous example of both infidelity but also the use of the mitochondrial sequencing to demonstrate at least a consistency with the historical record. Yeah. Yes.
So does that mean that these two living descendants are not, the transmitted descendants, but just related?
Yeah. So he would not. His mitochondrial genome came from his mother. So he could not pass that on to his descendants if he had any. I don't think he had any direct descendants. Yes, yeah, that's right. So you have to go to his mother's side of the family and then trace only through the females all the way down to the present. And the same argument goes here. I don't think he had any descendants, actually. So you'd have to find the closest common male ancestor, and then he would have received his Y chromosome from this person, maybe one generation removed, maybe. And then if this person might have received his Y chromosome from this common ancestor as well. If he had, that's right. And then if he had sons, then this person might be related to him through the sons and not through this common male ancestor. Okay. Okay. So I think we have a lot to cover today. So if you don't mind, I think we'll just keep going. Okay. So then now we're going to get into the topic of mitochondrial diseases. And we're going to, again, focus on mostly diseases that are caused by mutation in mitochondrial genes. But we'll also mention some that are caused by mutations in nuclear genes, and then diseases caused by somatic mutations. In other words, mutations that occur that you don't inherit, but which develop in your body as you grow older. So it turns out that there are these mutations in mitochondrial genomes are probably more common than previously appreciated. Part of the problem is up to now that it's been difficult to diagnose mitochondrial disorders. As we enter this era of whole genome sequencing and genotyping, these will be much more easily detected in the future. But it's already thought that maybe this many, 1 in 5,000, may become seriously ill. And this probably is also an underestimate of the importance of these disorders. Many mitochondrial diseases mimic other disorders or have diverse symptoms with varying severity. Common symptoms include fatigue, muscle weakness, neurological disorders, problems with vision, hearing, liver, heart, kidney, and bowels. So you can see that mitochondria can impact many, many different organs. Okay, so tissues that have high energy requirements are particularly sensitive to mitochondrial dysfunction, and this includes the retina of the eye, neurons in the brain, muscle, but also liver, kidney, and endocrine glands. And this just shows some of the places where where these organs are the focus of various diseases. Okay, so then as a clinician, then, where would you run into these? Well, you might find them if you're working in a clinic, a cancer clinic, or working in an epilepsy clinic, or any of these having to do with any of these sort of classes of disorders. The chances are you may run into patients in the future where the cause of the disorder has to do with mitochondria. And so, as clinicians, again, when I'm in a few minutes, I'm going to show you some examples of some very rare genetic variant diseases, and it's unlikely that you're going to see those more than once in a lifetime. But as clinicians in the future, you may actually run into many patients who don't fit in, for some reason are just a little different from the other patients that come to the clinic, and so mitochondria as a source of that pathology should be something in the back of your mind. Okay, so then why are mitochondrial diseases sort of hard to detect? And there are a couple of reasons for this. One reason is that each of us contains many mitochondria. Every cell contains hundreds to thousands of mitochondria. And these are not necessarily all genetically the same. For example, if a mitochondrial mutation occurred in your mother, that would affect only one of many mitochondria in their cells, in the mother's cells, and you may receive, that would be only a small fraction of the mitochondria that you inherit. So even if a mutation occurred in one generation, the next generation it may not have any effect because there are so many normal mitochondria that are present. But as generations go from generation to generation, as I'll explain in a minute, sometimes that balance can be upset. There can be just randomly more mutant mitochondria might end up in one descendant
than normal mitochondria. And so over time, the diseases can emerge. But the inheritance is complex. It's sort of hidden. It's not like Mendelian type of inheritance. So it's hard to detect. And then disease phenotypes are often modified by environmental factors or specific modifier genes, as we talked about last time, or different mitochondrial genetic backgrounds. Okay, these homoplasmy and heteroplasmy are really important concepts that you should. This will definitely be in the homework and probably on your board exam as well. So this is important. So cellular dysfunction in health often depends on the ratio of mitochondria harboring mutant or normal genes in the cell. There may be a threshold ratio at which the pathology appears to be different for different mutations. The presence of a single genetic type, normal or defective, is called homoplasmy. Okay, and later on I'm going to give you an example of a disease that is characterized, it's a mitochondrial disease, but usually doesn't appear unless almost all the mitochondria are defective. And for that reason, when you look at the mitochondria in the cells of these individuals, they're all almost always homoplastic. And this question often appears on board exams, so remember this. I'll tell you about the disease a little bit later. On the other hand, when you have a mixture of genetic mixture of mitochondria, that's called heteroplasmy. And many of the diseases I'm going to mention are characterized by this. In other words, not every single mitochondria has to be mutated in order for the disease to be seen. And mitochondrial diseases are often classified as homoplastic or heteroplastic based on the measured ratio of normal to mutant genes present in the cell. Okay, so this is just a picture of that. So this would be homoplasmy of a normal mitochondria. This would be homoplasmy of a defective mitochondria, and these would be examples of heteroplasmy. Okay, so remember these terms. Okay, and this is just another sort of diagram showing what I just mentioned, and that is that if your mother contains a mixture of mitochondria, some are dysfunctional, some are normal, you as an offspring can inherit any number of various mixtures of these. So under very rare conditions, you might get 100% normal mitochondria. On the other hand, if this is a substantial proportion of the mitochondria, then you're more likely to have a mixture. And then if you may, on the other hand, receive many more mutant mitochondria than normal mitochondria. And if you pass a sort of threshold, then you suddenly have this disease. Whereas if you have this situation, or a balance between mutant mitochondria and normal mitochondria, you don't have the disease. Okay, so the idea then is that diseases can emerge through a so-called bottleneck effect. So if if through several generations more or if in one generation more defective mitochondria are inherited, then in subsequent generations this percentage can increase, and that may be the cause of the appearance of a genetic disorder. Whereas if both the defective and normal mitochondria are inherited in equal proportion, then no disease is present, or maybe a mild form of the disease is present. Okay, so actually I should have shown this slide earlier, I think, but this is, I probably should have shown this earlier. If you make a picture of the pedigree for the inheritance of mitochondrial disease, you see this sort of pattern. So the mother's affected here. In principle, all of the children could be affected by the disorder, but only the females pass that disorder on to the descendants. So this person's affected as a male, but since the sperm do not, when this ovum is fertilized, the mitochondria in the sperm are lost. then the descendants of this male will not be affected. On the other hand, this mother will pass on defective mitochondria with a high probability to her descendants. Okay, so this is an example of a maternally transmitted effect. Okay, so the cellular pathways that are impacted by mitochondrial disorders are all the ones listed here, including electron transport, ATP synthesis, and mitochondrial biogenesis, replication. Okay, and then what I'm going to do then is, in the last part of this lecture, I'm going to go through 6 diseases in a little bit of detail, a slide for each one of these. And these are a list of that you often find in textbooks. And the ones I'm going to go over are the ones that be emphasized in genetics textbooks and often appear on examinations as well. Okay, so the Leber hereditary optic neuropathy, or LHON disorder, is a disorder that, as I mentioned before, is a classic disorder that it appears for mitochondria where almost all the mitochondria have this mutation. So this implies that if you have a mixture of normal and mutated mitochondria, you don't develop this. You have to lose almost all the mitochondria have this mutation, so that's largely homoplastic. Okay, so I would remember this as well. This would be a mitochondrial disorder that is characterized by being largely homoplastic. This is something that might appear in an exam, a board exam question. Okay, so this was discovered in 1871, named after its discoverer. And it affects mainly the retina, or the optic nerve. And what it causes is a blind spot here. And it's found fairly rarely. It affects 1 in 14,000 males. 60% are inherited from the mother. 40% are sporadic. 80 to 90% of cases are caused by mutations in mitochondrial, dehydrogenase, the NADH dehydrogenase subunit gene. So either the first subunit or the 4th subunit or the 6th subunit. And the common genetic mutations that cause this have been identified. So the mitochondrial DNA-associated Leigh syndrome is another famous disorder, and this is even more rare. And it is characterized by early onset progressive neurodegeneration, lack of motor control, dementia, seizures, generalized weakness, lack of muscle tone. You don't have to memorize all these, by the way, but it's important to remember this name and to know that later on there's also a form of this syndrome that's caused by mutations in chromosomal genes. And it's important to know that there are 2 forms of this. One mitochondrial DNA-based and the other chromosome-based. Okay, so, and most of the cases involve these mutations in ATP synthase subunit 6. And it tends to be heteroplastic. So even if a certain fraction of the mitochondria carry this mutation, you end up with this disorder. And there are different sort of in an MRI scan. There are different sort of signatures in the brain that are detected. So these white areas right here are abnormal signals, and they are often seen in this Leigh syndrome. Okay. And I'll mention later on there are autosomal recessive and X-linked forms of this syndrome as well. Okay, so another very famous syndrome is this mitochondrial myopathy, encephalopathy, lactic acid acidosis, and stroke-like episodes, MELAS. Acronyms are very useful in this area, in this field. Okay. This has a fairly high frequency in northern Finland, and also northern and Sweden as well. The characteristics are muscle weakness and pain, seizures, elevated levels of lactic acid in the cerebrospinal fluid, and stroke-like episodes before age 40. And then in an MRI scan, there are also signature areas this is where there's atrophy of the brain here, and probably the cause of the stroke-like episodes. It also can present with diabetes and deafness, and also this chronic progressive external I can not pronounce this, plegia, or CPEO. And what this is a symptom that's seen in a lot of these disorders, where the eyes become, you can't move the eyes, they become rigid. And then often the muscles of the eyelids become weak, and you develop drooping eyelids as well. So people, when they're developing the disease, tend to hold their head back like this and look, because their eyelids, they can't hold their eyelids up. Okay, and this is one of the disorders that is the result of these mutations in the tRNA that involve the inability to add this taurine residue right here. And when this occurs, then proteins that are particularly rich in leucine, there should be only one leucine, for example, cytochrome oxidase, are differentially affected. The ability to add leucine is not completely lost, but it becomes less effective. So proteins that are particularly rich in leucine residues are not synthesized in adequate amounts, and that this disorder. Okay, another famous one, myoclonic epilepsy with ragged red fibers, MERFF, birth. This is also extremely rare, 1 in 100,000. The characteristics include progressive myoclonic epilepsy and ataxia, and the presence of these ragged red fibers. So this is another term that you should probably remember, and this may also appear on board exams. And what these are defective mitochondria that are visualized by a special stain called the Gomorrhi trichrome staining, and you can see the staining right here. So these are the so-called ragged fibers that are characteristic of this disorder. And here, this is also caused by a defect in the chemical modification of the tRNA. And again, it affects this first position that is needed to bind to the wobble position in the codon, in the mRNA. Okay. And the NADPH dehydrogenase and cytochrome oxidase are often affected by this mutation as well. And then the Kearns-Sayre syndrome, KSS, is here's an example of this is characterized by this chronic progressive external ophthalmoplegia. I can say it now, CPEO. And what that is, again, one characteristic of this is the inability to hold the eyelids open. So this shows the progression of the disorder from 1998 to 2004, and you can see the ability to open the eyelids is reduced. And then, in addition, there are also often problems with the retina as well. And it's hard to see, but if you, when you look at these slides at home, you should see brown speckles covering the retina here, and that's a characteristic sign. And then there are also ragged red fibers that can be seen in the muscle tissue. Okay, and then the last one I'm going to talk about, the last mitochondrial DNA disorder, is NARP. This is also extremely rare. Neuropathy, ataxia, retinitis pigmentosa. And here, this is also characterized by ragged fibers in muscles, and then by high lactic acid in the brain. And this is detected by magnetic resonance spectroscopy. Okay. Okay, so then this is that diagram I promised I'd show you, showing this is the mitochondrial genome, showing the positions of various genes, and these are the diseases that I just mentioned, and you can see that they're the sites of the mutation that cause these various disorders. Okay, and then I'm just going to go very quickly through mitochondrial diseases that are caused by mutation in nuclear genes. So another form of Leigh syndrome is caused by autosomal recessive or X chromosome linked forms of mutations. And these are usually involve proteins in the oxidative phosphorylation pathway. And of particular interest, there's a population in Quebec where this is particularly frequent. So this is something that, as clinicians, that you should keep in mind in general. That, for different disorders in the general population, the frequency of a disorder gene may be very, very low. But within specific populations, last week we talked about the Ashkenazi Jewish population, for example. There were cystic fibrosis mutations. The mutations that cause cystic fibrosis are fairly common. This is true of mitochondrial deficits as well. So when you're dealing with patients, it's very important to know something about their family history and what population they may have come from, because that may change the range of possible diagnoses that you would consider. Okay, so this is another rare disorder, mitochondrial neurogastrointestinal encephalopathy. And this has to do with, is caused by mutations in an enzyme that is important for the metabolism of thymidine. And when this enzyme is defective, this builds up in the brain and in other tissues and causes damage. And the mutation is caused by mutations in this TYMP gene. And then another famous example is the Alpers syndrome, or Alpers-Huttenlocher syndrome. And this is caused by mutations in the DNA polymerase gamma protein, which is shown here. And this is also famous because it has a very high, the frequency of the mutations are the highest in people who live in this island right here off the coast of Norway, the Faroe Islands. And so this is another example where a specific population can have a very much higher frequency of a disorder gene than's found in the general population. Okay, and then in addition to the disorders I've mentioned up till now, the mitochondrial gene-based ones and the genomic chromosome-based disorders, are ones that you usually inherit from your parents. But in addition to that, there are mutations that occur somatically. In other words, as we grow older and cells divide, sometimes mutations occur. And these have been detected both for mutations in the gamma polymerase, DNA polymerase, but also in Alzheimer's disease and Parkinson's disease. It's been found that, for example, 75-year-olds with AD contain more of de novo mutations within mitochondrial genome than normal. And so it's possible that this has some connection with that disorder as well. Okay, and then the very last slide I'm going to show you talks about the possible germline therapy for primary mitochondrial diseases. And this is also something that's been in the news just very recently. That it's possible through genetic engineering to make babies that have three parents. And the way this works is that, suppose you have a mother with a mitochondrial disorder wants to have a healthy baby. But she has a mitochondrial disorder, so any child that she is conceived normally will receive all those mutant mitochondria. So what's to be done? The idea then is you take an egg from a normal woman with normal mitochondria, and you remove that nucleus. Right, this is that right here. And then you take the nucleus from this woman who wants the baby and put that into this cell, okay? And now you've replaced the nucleus and hopefully not brought any mutant mitochondria with that nucleus in here. And then when this is fertilized, a normal baby can be produced. So this baby inherits all its genomic DNA came from, nuclear DNA came from the mother and from the father, the donor of the sperm. But the mitochondria came from a third individual. So this, in theory then, this baby has three parents. Okay. And there were actually, this procedure was carried out sort of on an experimental basis in the around 2000. And there are a couple of babies that have been born using this process, and they apparently normal. They grow up normal, and they're free of the mitochondrial disease. But this was quickly shut down, at least in the United States, by the FDA, Federal Food and Drug Administration, because it was considered too risky and just experimental. And there were a lot of people who raised sort of ethical questions about this as well. They were worried that the child would grow up with a psychological complex or something because they had two mothers and one father. But just this year, the FDA has begun to consider this again. So 15 years after the first babies were born, the FDA is right now reviewing this. And it may be that in the near future this will become a common procedure in the United States as well. There are other techniques, and instead of transplanting the nucleus, it's also possible, apparently, to take the metaphase spindle from a cell that's undergoing duplication. So you have the metaphase spindle with the microtubules and all the metaphase chromosomes. And it's possible to actually transplant that into the donor cell instead of the whole nucleus. And apparently that technique works as well. So this is something that probably within the next year, probably next year, there'll be a decision on this. So this is a procedure that may become available again. And so among your patients in the future, you may find some that have three parents, genetically three parents. Okay, so I think that's it for what I want to cover today. And I have some additional slides that you can look at when you look at the PPT, and these are just for your reference. They won't be included in the homework or the final exam. Okay. So any more questions? Or? That can give transmit them to the children, to the next generation, yeah. But it's different from the X chromosome inheritance in that both males and females with equal probability essentially are receiving the mitochondria. So with the X-linked inheritance, if there's a mutation on one of the two X chromosomes, a male who receives the normal chromosome will be normal. Yeah, and whereas if a male that receives a mutated X chromosome will have the disorder, whereas females receive both chromosomes, and it depends on the random inactivation of the X chromosome, the proportion of normal to inactivated chromosome X's that determines the severity of the disorder. Yeah. Okay. So have we decided on a place to meet? Where do you want? Should be in the other class, or do we know that classroom's free, for example? Where are we going to meet next? Where's the lab room? Number 5. Okay, I can find out where it is. And is there a place to sit down like this? Oh, okay. You have what? Oh, you have a seat. Okay, so I assume that's going to be that should be open. So okay, so we'll meet. I'll find out where that is. I'll bring my slides. You bring your questions. And we'll have a discussion for a couple hours. And if that's not sufficient to answer all the questions, we'll schedule another one after that. So that'll be next Wednesday at 1:30. Is that right?